um, in the interest of time. So thank you everybody for joining. Sorry for the last minute change to remote presentation, but we look forward to two really great talks today. Our New Faces talk is by Dr. Engi Attia. Dr. Attia is an assistant professor of medicine in the Division of Pulmonary Critical Care and Sleep Medicine and in the Department of Global Health at the UW. She's also co-director for the CFAR BPIN, the Biomarkers, Prevention, and Interventions for HIV-Associated Malignancies and NCDs core. Her research examines chronic lung disease among people with HIV, including K23 and R03 grants from the National Heart, Lung, and Blood Institute to examine mechanisms, phenotypes, endotypes, and risk factors of chronic lung disease in adolescents with HIV in Nairobi, Kenya. She's also supported by a pediatric HIV/AIDS cohort study and the Fred Hutch UW CFAR to compare chronic lung disease in US and Kenyan youth, specifically investigating the relationship of HIV and air pollution to lung function. And this morning, Dr. Attia will address the question, can metabolomics and quantitative CT improve endotyping and phenotyping of chronic lung disease among youth with HIV? So with no further ado, let me hand it off to Dr. Attia.
Thank you so much for that introduction. I really appreciate the opportunity to share with you all the work that we've been doing in Nairobi. I want to just start by giving an outline of what to expect. We'll talk about some background in terms of chronic lung disease and how it pertains to HIV and children and adolescents, which I'll intermittently refer to as youth living with HIV. And then we'll talk about chronic lung disease, what is known about chronic lung disease phenotypes and endotypes, the research that we've got going at the Coptic Hope Center in Nairobi, and then next steps, future directions, and sum it up. So to get started, I just want to highlight that globally, chronic lung disease accounts for 7% of deaths and 4% of disability-adjusted life years every year. And then when you start thinking about this more granularly, in the context of HIV, and specifically in Kenya, you can see that chronic lung disease in the blue squares here and HIV both contribute a substantial number of disability-adjusted life years, but HIV really seems to outshine chronic lung disease in terms of those contributions. But when you start thinking about disability differently and think about long-term disability and years lived with disability, those contributions start becoming much more similar, which really leads to the understanding of why there's been a shift recently in HIV-related pulmonary complications toward chronic lung disease across the lifespan, but we're going to focus on youth with HIV today. Among youth with HIV, it's been documented that a majority have at least one chronic respiratory symptom, and many have low oxygen levels after minimal exercise, like going for a brief walk. And radiographic abnormalities are common on CT and chest X-ray. And in sub-Saharan African countries, impaired lung function has been noted in up to 45% of those who have spirometry or more comprehensive lung function tested, and up to about a third in one study in the US. I want to take a moment to try to convince you all that this is important, and why I am really focusing on studying chronic lung disease in youth with HIV, particularly those with perinatally acquired HIV. So in the general population, it's known that early life factors are linked with impaired lung function later in life. And some of these factors include fetal oxygen and nutrient delivery, low weight at birth and during childhood, malnutrition or undernutrition, inhaled pollutants. And in this figure, you can see how those exposures and others like TB and pulmonary infections and occupational exposures are present across the entire lifespan. And in the top part of the figure, I want to call your attention to the substantial amount of lung growth and development that occurs during the age of adolescence from 10 to 19 years old, before there's a peak and plateau in the early 20s, followed by, unfortunately, a normal decline in lung function as we age. And an additional unique consideration in terms of young people living with HIV is that many of them have perinatally acquired HIV. So they've had long-standing HIV during critical periods of lung and immune development. And this has been found to be associated with greater prevalence of end organ injury in terms of other organs like the cardiovascular system and the neurologic and central nervous system. And this might be driven by chronic inflammation, immune activation, and endothelial activation. And I think that you all will be familiar with also why specifically youth are important to study in this context. So in 2022, over 1.5 million youth aged 10 to 19 years old were living with HIV, and there were about 140,000 new infections that year alone in this demographic. And then in a decade, from 2005 to 2016, the number of youth living with HIV increased by 30%, which is the opposite direction of the gains that have been made in other age groups. And the dying due to HIV-related illnesses tripled. And most of these youth live in sub-Saharan Africa, and they account for most of the new infections. And if things keep going as they are, we can anticipate having more than 1 million new additional youth living with HIV by 2030. So in our early work, we studied a group of adolescents and adults at the Coptic Hope Center in Nairobi. And we found that accounting for CD4, BMI, severity of HIV at diagnosis, TB, and inhaled exposures, adolescent age was associated with a 3-fold risk of abnormal lung function among people with HIV. And in a group in South Africa, they found that lung growth lags in youth with HIV, even if they start ART early. And in this case, they initiated ART at 4 years of age. So I hope that I've convinced you that it's important to learn about what's going on with these children in terms of their lung health. And now we'll delve into what we know about chronic lung disease phenotypes and endotypes in these youth. Specifically, I want to highlight that phenotypes represent observable characteristics and things like symptoms and physical exam, and can be derived from commonly available clinical data like lung function testing, such as spirometry, chest imaging, lab values, 6-minute walk test, and a history that you would obtain about exposures and risk factors. The sentinel work in this field really was published in 2012 and highlighted that there was delayed diagnosis of perinatal acquisition of HIV in these adolescents who had chronic lung disease. And they found a substantial number of symptoms and recent respiratory infections and low oxygen saturations, and 41% had poor lung function. And and 55% had what's called mosaic attenuation on high resolution CT, which together with the low lung function might suggest a lung disease called obliterative bronchiolitis. And here is a representative image where you have dark areas of CT that have higher aeration or more air and lower density, and then these high density regions that are right next to it. And that's how we define mosaic attenuation. Obliterative bronchiolitis is unfortunately a disease that is largely irreversible and causes destruction of the small airways, likely in the setting of inflammation, and can be seen in other states like stem cell transplant and lung transplant rejection, etc. In our group, we similarly found that adolescents had a high proportion of respiratory symptoms, and this was significantly higher than what we saw among the adults, despite the fact that adolescents had better HIV control and fewer exposures and comparable prior respiratory infections. And they also had a high prevalence of abnormal spirometry patterns that could help us understand some phenotypes. So they had low FEV1 to FVC ratios, which represent airflow limitation or obstruction. And then they had a low FEV1 and low FVC compared to adults as well. And those in the context of a normal FEV1 to FVC ratio are representative of a category of abnormal lung function referred to as PRISM, or preserved ratio impaired spirometry, which has been associated with poor outcomes in the general population, including increased mortality and respiratory deaths, as well as hospitalizations and exacerbations. There has been a study using quantitative CT analysis looking at those with obliterative bronchiolitis, also known as bronchiolitis obliterans, in perinatally HIV-infected adolescents living in South Africa. And they used a process called semi-automated lung segmentation to look at CTs from 78 youth with HIV and 16 uninfected youth. And they found that those with HIV had higher lung density, more ventilation, heterogeneity, and more regions that had high attenuation and low attenuation. So the kind of darker and lighter regions that we were talking about. Sorry, the lighter and darker, respectively. So these low attenuation regions and ventilation heterogeneity really correlated with a lower FEV1 and FVC, suggesting airflow limitation again, and maybe obliterative bronchiolitis, but they also detected a high percentage of high attenuation, so the brighter regions than the low attenuation areas, which you can see in the figure here represented as the green regions, with the lower attenuation regions being the blue. And so these high attenuation regions have been documented to reflect higher inflammation among people, or I should say adults with HIV. The pathologic correlate hasn't been established fully in youth with HIV, but it's presumed that it represents the same process. And so the phenotypes of chronic lung disease remain really poorly understood, and they can span from airway predominant to parenchymal and vascular predominant. And as you can see here, the airway predominant ones would include things like obliterative bronchiolitis and bronchiectasis, asthma COPD overlap, while at the opposite spectrum, where we see kind of those higher density areas, you might think about interstitial lung disease and fibrosis and LIP, as well as things like pulmonary hypertension. And then kind of in the middle, because we don't know where they fall and how they impact the lungs sufficiently, we think about post-TB lung disease and recurrent infections. Then when we shift gears and think about endotypes, it's important to think about these distinctly from clinical phenotypes, but they still do link the expression of disease to the underlying mechanisms of disease. So endotypes can be defined as a subtype of disease that's defined functionally and pathologically by a molecular mechanism or by a treatment response. And the heterogeneity of chronic lung disease, as well as all of the exposures and factors that can contribute to it, really lend itself to the study of endotypes here. And you can use multiomics, and specifically I'll share some metabolomics and immunologic assays with you. And it's important to keep in mind that distinct endotypes may coexist, and there may be more than one to explain someone's abnormal lung function and chronic lung disease. And the associated clinical features reflect differing molecular causes and potential responses to therapy. We know very little about endotypes, again, of chronic lung disease among youth with HIV. Among those adolescents who had early ART initiation, they had better lung function. And similarly, in school-age children with perinatally acquired HIV who initiated antiretroviral therapy in infancy, they had lung function near population norms. So in terms of a treatment response, it seems that antiretroviral therapy has a beneficial response, as would be expected when initiated early. And there is a single randomized clinical trial in this field of weekly azithromycin for 48 weeks. Unfortunately, in terms of treatment response, there was no difference in the FEV1 or lung function, but there was a decrease in respiratory exacerbations in those on azithromycin. As expected, they did demonstrate some azithromycin resistance on nasal and sputum samples, but among those who had preserved bacterial diversity and decreased Haemophilus influenza and Moraxella, they did find that there was improved lung function. And there's a question of how this interacts with administering azithromycin. In our group, when we looked at Kenyan and US youth with perinatally acquired HIV, we found that immune imbalance and activation, as measured by a higher CD4 and a lower CD4 CD8, were associated with lower lung function, even in the setting of CD4 preservation and reconstitution. And then in a small cohort of our Kenyan youth, we also found that individual biomarkers, as well as factors of biomarkers that represented endothelial activation, innate immune activation, and inflammation, were associated with airflow limitation and obstruction among adolescents with HIV. And then when thinking more specifically about the obliterative bronchiolitis phenotype, we found that immune activation measured by soluble CD14 was associated specifically with that endotype or that phenotype. In terms of metabolomic profiling, many of you, I'm sure, have heard of it as a tool to identify signatures in diseases, and it's been used in some lung diseases recently and has found unique pathways, including pyrimidine, lipid, fatty acid metabolism, as well as glycolysis, mitochondrial oxidation, and the TCA cycle. But metabolomic profiling, to our knowledge, hasn't been performed among youth with HIV who have chronic lung disease. So that leads me to talking a little bit about the research that we've been doing at the Hope Center. And this study is funded by the MyK23, which finished recently. And so data analyses are in full force. And thankfully, I've also had the privilege of having some sub-studies that allow things like the quantitative CT and metabolomics be funded by the CFAR and FACS and UW Bridge funding. And I'm incredibly grateful for those opportunities. So for the overarching objectives of the research, we are comparing phenotypes and risk factors of chronic lung disease among youth with and without HIV. And our goal is really to determine whether HIV is associated with chronic lung disease and lower growth of lung function over time, and then determine whether endotypes identified by immunologic assays, as well as metabolomic profiling, are associated with increased risk of chronic lung disease at baseline, as well as diminished lung function over time. I won't share with you any of the lung function over time data, as those are still in the works, but hopefully they'll be ready for the next time I'm able to talk with many of you. So a little bit about our site. The Coptic Hope Center for Infectious Diseases was founded by the University of Washington Department of Global Health, led by Grace John Stewart and Michael Chung, 20 years ago at this point, and it is going strong and is a wonderful resource and one of the biggest HIV treatment centers in Kenya. We enrolled 165 youth with HIV and 170 HIV uninfected youth, and excluded any who had acute respiratory infection, severe illness, recent TB, or pregnancy, as that can impact your lung function. And our key study procedures included spirometry before and after bronchodilator, and courtesy of COVID, that data collection spanned 4 or 5 years, longer than the 2 years we'd anticipated. And we collected high-res CTs, which were interpreted visually as well as with quantitative metrics, and serum for biomarkers and metabolite analysis. And then we are also doing some personal level and satellite level air pollution collection. So a few things I want to highlight between those with and without HIV really are that the CD4, CD8 ratio is quite low among those with HIV, but normal in those without HIV. And prior pneumonia is similar, but prior TB is more common among those with HIV. And you can see also that the BMI and height for HZ scores are substantially lower among those with HIV. Nonetheless, inhaled exposures seem to be reasonably comparable. And to our surprise, respiratory symptoms, and I'll show you later, lung function was abnormal in a substantial majority of those with and without HIV. Nonetheless, youth with HIV still had significantly lower lung function as measured by FVC and FEV1. You can see here, though, that the ratio of the FEV1 and FVC were pretty comparable between groups. One of the main findings of our study is that HIV is independently associated with any abnormal spirometry pattern, as you can see here, as well as the PRISM pattern that I was referring to earlier. And this really pins down why we're going to focus on those with HIV in our studies. And another piece that really has driven us to explore things like quantitative CT and metabolomics in trying to characterize chronic lung disease in this cohort is, as you can see, abnormalities are incredibly common and heterogeneous and overlapping. So among those who have abnormal CTs, and that's 91 of 152 with complete data, nodules are common, mosaic attenuation is common, bronchiectasis and lymphadenopathy and scarring is less common, but most of these findings are duplicative in one person. And then while abnormal spirometry tends to be a little less heterogeneous, how it relates to CT abnormalities has also been a bit of a challenge in terms of understanding. And then respiratory symptoms are quite nonspecific, but also very heterogeneous and overlapping and multiply present. And just as an example here, I just wanted to show you one of the more impressive CT scans from our cohort that shows how there are, in fact, multiple abnormalities happening in one person. So here you see these high density regions and the lower density regions by each other, representing mosaic attenuation and bronchiectasis with bigger airways and surrounding bronchial wall thickening, as well as some cystic and emphysematous changes and some nodules and areas of ground glass. So we are working with a group at the University of Michigan to look at quantitative CT metrics to help us more objectively define what's going on in the lungs of these youth. And we are using a process called parametric response mapping, which takes into account inspiration and expiration data. And although the color scheme is different compared to the image I showed you earlier from the South African group, what I want to call your attention to is this purple or magenta color, which is the high density area that might represent parenchymal disease. In our case, green is normal in contrast to the earlier images that I was showing you. Importantly, the quantitative CT metrics, but not the qualitative findings, which I won't show you the details of now, differ by abnormal spirometry among youth with HIV. And so in the group with HIV here, you can see that those who have abnormal spirometry have lower lung volumes. They have lower Hounsfield units or density on expiration and inspiration. And then the green areas or the normal lung areas are fewer in those with abnormal spirometry, and the parenchymal disease areas are higher. So, this helps us really objectively start to define what's going on in these kids' lungs. But it doesn't quite yet get us to the granularity we need to understand some of the phenotypes that might be present. Then shifting gears a little bit to the endotypes piece. So we, on a bigger scale now, looked at the biomarkers and immunoassays that we had looked at in the smaller scale and found similar findings. So soluble CD14, soluble ICAM1, and soluble VCAM1 that here clustered as a factor that represented endothelial activation and immune activation were associated with any abnormal spirometry as well as the prism pattern among those with HIV, but not those without HIV. You can see here, though, that chronic inflammation was associated with abnormal spirometry in both those with and without HIV, which is not unexpected. And then when we looked at our metabolomics, this is kind of new, hot off the press data, we found that there were some distinct metabolic profiles of oxidative stress and pyrimidine metabolism that were associated with respiratory abnormalities and low lung function defined as FEV1. And interestingly, it's been shown that oxidative stress is associated with some mutations in pyrimidine analogs in other diseases, but not specifically in lung disease. And so in conclusion, we found that HIV was associated independently with lower lung function and greater risk of abnormal spirometry. And among those with HIV, there really was this substantial heterogeneity and overlap in manifestations of chronic lung disease that made it challenging to identify clinical subtypes. And so the high density areas of parenchymal disease on quantitative CT are kind of a good first step for objectively identifying phenotypes of chronic lung disease. And then we also found endotypes with metabolic pathways of oxidative stress and pyrimidine metabolism and endothelial and immune activation associated with respiratory abnormalities. In terms of next steps, we are working with the University of Michigan group to do some personalized attenuation thresholds and deep learning models to further quantify our chest CTs. And the longitudinal data that I told you about is in the works, and we hope to add some endotyping with proteomics, epigenetics, and microbiomes. And the summary is basically what I mentioned before, but the take home really is that chronic lung disease is an important complication of HIV and that the manifestations are heterogeneous and overlapping. And it's challenging but exciting to try to identify clinical phenotypes and endotypes. So many thanks to everyone who has supported me along the way. And I'd like to just acknowledge all of the support that I've received from different institutions and open it up for a few questions if we have a couple minutes.
So we're almost at time, but thank you very much for that wonderful presentation, Engi. And if we probably can take one question, if there's.
Otherwise, I'm happy to answer questions by email or schedule a talk or call another time.
Thank you very much. Maybe if I can throw out one quick question. I was wondering whether there's been any histological correlates with the specific findings, clinical lung findings, the nodules, the high densities on CT and so forth. Is that correlated with any particular histological findings? And does it have any continuum between the high density, the nodules, and the different phenotypes by CT?
That's an excellent question. That has not been done yet. I think the challenge really has been to really implement those kinds of invasive procedures in a resource limited setting, especially among young people. Some of the work that we are looking at doing would be to do some less invasive sampling that might give us some ideas. So do induced sputum and do some measurements on that and exhaled condensates, things that kind of reflect what's going on in the lungs without specifically biopsying them or doing a bronchoalveolar lavage. But someday I may gather up the nerve to do that.
Well, thank you so much for a wonderful presentation. I have tons of little questions to ask you, but we'll have to do it offline, and hopefully
Sounds
others
great.
will also approach Engi with their questions. But
Yes, I'd love to.
we should probably move on to Nyari. So our main speaker is Dr. Nyari Chigorimbo-Tsikiwa, who is joining us from Cape Town, South Africa, where she has recently joined the International Center for Genetic Engineering and Biotechnology, the ICGEB, which is an intergovernmental organization with research laboratories in Italy, India, and South Africa. The Cape Town part of the ICGEB is based in the University of Cape Town, where Dr. Chigorimbo-Tsikiwa received her PhD and was a senior research fellow in the Division of Medical Virology. As a postdoc, Nyari was part of the multidisciplinary team developing Africa's first anti-malarial drug, MMV-048, but her passion for viral immunology has led her to study host virus interaction for HIV-1. And her interest is to understand the cellular and molecular mechanisms that impact HIV susceptibility in humans on mucosal surfaces, particularly in the male genital tract. And she's currently focused on using immunologic and multi-omic approaches to better understand the diverse myeloid cell populations in the genital tract and their interactions with viruses as a means to dissect the molecular mechanisms underlying HIV susceptibility. And that leads to the title of her talk, Host Factors Influencing HIV Infection in the Male Genital Tract. Nyari, take it away, and thank you for joining us at such a late hour in South Africa.
Thank you, Jay, and thank you to you guys at the CFAR for inviting me to share some of the work that we've been doing in Cape Town, South Africa, looking at various things in the male genital tract. But I'm really sorry I couldn't make it in person. I was really looking forward to sharing this work with you guys in person, but unfortunately, I will have to do that another time. But let me get straight into it, and we'll change tack a little bit from more clinical based kind of study to a more biological mechanistic look at host factors that influence HIV in the male genital tract. And as Jay said, I am based at the ICGEB. Most people don't know it, so I thought I might just share a few slides about who we are and what we're doing. You can see my slides, is that correct, Jay?
Yes, no, very nicely.
Okay, awesome. Yes, let's get this moving. Okay, so this is where I live in Cape Town, the most beautiful place in the world. And the ICGB is an intergovernmental organization that is a research organization that is kind of follows the model of the United Nations. And what our aim is to use science for development at the ICGB. So our headquarters is in Trieste, Italy. But we have three components with the three research centers, one based there in Trieste and a really big one in India, and where I'm based at the University of Cape Town, which is in the Institute of Molecular Diseases and Molecular Medicine. So we're based at the University of Cape Town, but we're not part of the University of Cape Town, but I've lived all my life and studied at the University of Cape Town. And so all this work, in fact, that I'll be sharing with you was conducted when I was there at Cape Town. And so I don't think I need to highlight to anyone in this audience here the global issue that we have with HIV. But the major thing that I would like to share here is that sub-Saharan Africa is disproportionately affected by the global HIV pandemic. What we find actually is that 2/3 of people living with HIV live in the African region, which has massive ramifications for us, and some of which I'll speak about later, which particularly refer to biotropism and some of these host genetics and those kinds of things. But we'll get to that in a minute. But the main reason why, is that a comment? Oh. Okay. So globally, most of HIV infections are acquired by males across the penile tissue. And whilst HIV acquisition leading to infection has really been intensively studied in the female genital tract, very little is known about the viral acquisition events that take place in the male genitalia. And so understanding the biological factors that increase the risk of acquiring HIV in the male genital tract, we think are important really in the design of therapeutics for HIV prevention, but not only for prevention, but also in trying to understand what is happening in the mucosa, with particular to trying to design HIV cure studies as well, because we think it's an important site. So there are various things that we know about the male genital tract, and in particular in the when we look in the penis, there are various correlates of HIV susceptibility and protection that we've seen. We've seen from various studies that medical male circumcision, for example, reduces HIV infection. Whilst our studies really haven't focused on trying to understand that, we've used it as a source of tissue to really interrogate some of the questions that we have. And so we do understand that male physiology does play a role in understanding the HIV protection. And as I mentioned, we do understand that different regions within the male genital tract, even on the penis itself, have different correlates of susceptibility. So for example, the inner foreskin, which covers the glands in a non-erect penis, is known to be more readily infectable to HIV in ex vivo studies, whereas the outer foreskin isn't. And the mechanisms for that are quite interesting. I think Maria Lemos, who's in the room, will have some comments about that. But really, some thoughts were about keratinization, but I think most studies have shown that keratinization is really not the one factor. But what we do know for sure is that the foreskin and the male genital tract has a plethora of HIV susceptible cells. And so these HIV susceptible cells and how they are present and how they're moving in and out of the male genital tract, we think is an important question to answer. And so we have focused our studies on the foreskin, and the reason for that is that studies have shown that the size of the foreskin has a direct correlation to risk of HIV infection. In fact, these are studies that came out of Rakai, and I think Jessica Proger and Rupert Cowell were responsible for these. So we do know that the foreskin removal reduces the risk of HIV infection, and a lot of this was seen in the randomized clinical trials that were done also in Rakai that showed up to a 60% reduction in HIV acquisition. And so the foreskin is an immunologically competent tissue that is full of all these incredible immune cells that are there. And so we know that it has high densities of these HIV susceptible cells, which are various, from the CD4 cells as well as Langerhans cells, and myeloid cells, which we've come to understand a bit more about their biology in relation to HIV acquisition recently. And as I mentioned, we know that the removal of the foreskin really decreases the chances of acquiring HIV. And so the current thinking that we have been looking at has just really been that there are various factors that impact the host with regards to looking at HIV susceptibility. So some of these factors are genetic factors. So we understand that the genotype, for example, with the mutation in the CFAR5 gene confers resistance to acquiring HIV. And we know that the presence of STIs such as syphilis and gonorrhea increases the risk of STIs. And a lot of that information is thought to be due to the lesions that some of these STIs form. And so these lesions result in depleted barrier function, and which results in the eased entry of HIV in subsequent exposures. And we also understand that there are various molecular factors. And a lot of these molecular factors, particularly the role of various cytokines, have been elucidated in the female genital tract. Various studies have shown that a lot of pro-inflammatory cytokines, IL-8, are associated with increased risk of HIV acquisition. And we wanted to also have a look and see what happens in the male foreskin. And there have been studies that have tried to look at this, looking at the cytokines as well, which I think we looked at cyto brushes that were taken from swabs within the penile urethra, quite a painful procedure, I would imagine. But from those studies, really, we did see that there were cytokines that are involved that increase HIV infection. So there are all these molecular factors that also do that. And then there are cellular factors, which we spoke about, with HIV target cells, and microbiome dysbiosis, which is work that has been done quite extensively by Jessica Proger, as well as various other people, really has highlighted the role that the microbiome plays in increasing HIV acquisition with these anaerobic bacteria, really resulting in these increased risk to HIV acquisition. And so with that, we wanted to, with all this knowledge that is there about all these things that sort of play a role, we wanted to take a holistic approach to kind of understand the different players that contribute to this HIV susceptible state that is present in the male genital tract. And why this is important is that we know that not all sexual encounters in the male genital tract result in HIV acquisition. In fact, most of them are not. In studies that have been done by Patel et al. In the systematic review, in which they were talking about, how many sexual acts it takes for HIV acquisition to take place. We've seen actually that most of these incidents are protective. And so the question is, what differs between those protective, times when the immune system is able to deal with the virus, and when the immune system isn't? So what are those correlates of protection, if you would say? And so I think the understanding really is that it's a continuum. Is it one factor, or is it a combination of different ones? And what is it that could confer this antiviral state, or what is it that results in this protection to happen? And so we know there are other factors, like I mentioned previously, like asymptomatic STIs, as well as STIs, as well as microbiome dysbiosis. But so what we decided to do was to really look and try and evaluate what the different associations with regards to the cell states and phenotypes that we find within the male genital tract in the mucosa itself. And also look at the impact of viral factors, because why viral factors are important, we think, is that the viruses that impact sub-Saharan Africa are predominantly subtype C, which is actually responsible for the majority of HIV infections. It is understudied, as most studies that have looked at HIV infection have really focused on subtype B. And we know from the work done in COVID that, in fact, the impact of subtypes is really important, particularly when looking at acquisition and also looking at disease progression and those kinds of things. And so for us in Africa, we think looking at viral tropism is an important question. And so we'll speak a little bit about the work that we started trying to investigate that. And then trying to understand what it is about these inducers of susceptibility are. So what is it? What are the mechanisms that these inducers of susceptibility really use to increase the susceptibility? And so to do that, we started looking at the impact of asymptomatic STIs on as one of the molecular factors that can impact HIV susceptibility. And in this case, we looked at asymptomatic STIs, and we looked at asymptomatic STIs because the current knowledge, the current protocol is you do not treat currently a lot of individuals who have STIs actually do not present. So chlamydia, 50% of men who have chlamydia do not present clinically. And so there is a lot of subclinical incidence of STIs, and the non-lesion STIs therefore are quite understudied in looking at what their role is in regards to increasing HIV infection. So we know, for example, that HPV does increase the risk, but what about the role of some of these asymptomatic STIs? And so So we looked at the effect of asymptomatic STIs on the foreskin proteome, and the reason why we looked at the proteome was really to try and look at the molecules themselves, and proteins are the effector molecules that are involved in, in these biochemical processes. And so we wanted to understand what is happening on the molecular level, on the protein level, with the impact of asymptomatic STIs. And we took a metaproteomic approach, which allowed us to not only look at the changes that were taking place in the host proteome, but also to look at the changes that were taking place in the microbial proteome as well. And so this work has been published, so I will not speak too much about it, but I'll just highlight some of the things that have caused us to ask further questions. And so what we did in the study is it was a sub-study in a larger study in which we looked at the presence of asymptomatic STI in a cohort of adolescent males who were receiving voluntary medical male circumcision. And we tested for several asymptomatic STIs that were detected by PCR in the first pass urine, and these were gonorrhea, chlamydia, and we also looked at Trichomonas vaginalis and Mycoplasma genitalium. We also investigated HSV, but we did not find a high incidence of HSV in our cohort. We found a prevalence of about 14% of individuals in our cohort that were presenting with asymptomatic STIs. And of those individuals, what we did is we took 16 individuals who were presenting with asymptomatic STIs, and we age matched them to 10 individuals who were not, and we conducted bottom-up LC-MS metaproteomic analysis of these foreskins. And those of you who are familiar with proteomics understand that everyone hates to work with skin, very difficult to work with. And so this was quite an interesting approach to take for us. And so using this LC-MS analysis, we followed this novel metanova pipeline in collaboration with other researchers at the University of Cape Town, Nikki Mulder, particularly Jonathan Blackburn. And what we did there was to design a protein database that included microbial proteins as well. And so using this really specialized database, we were able to identify bacterial and both proteins, as I mentioned. And then we conducted relative quantification of the proteins. And it's lovely to have a long list of proteins that are upregulated and downregulated, but what does it mean to have all these different proteins? And so what we did was we conducted functional gene analysis using protein enrichment analysis to really understand what phenotypes are conferred by these proteins that are differentially expressed. And for this, we use the TopGo gene ontology analysis in FGNet, which is a package that you can use in R and to conduct a gene ontology enrichment analysis. And what we found was, indeed, asymptomatic STI significantly alter the foreskin proteome. And when we went into the study, we had hypothesized that we would find differences in the foreskin proteome due to compartment. We thought that the inner foreskin would be quite distinct from the outer foreskin, but our data did not show that. In fact, all the differences that we saw were driven by the presence of an STI. So in this heat map, what I'm showing you here is an unsupervised hierarchical clustering showing the differential protein expression, showing fold changes in expression of the proteins in this proteome. And unfortunately, it is quite small, but what you can see right at the top there is that the clusters that we see really were clustering by STI, which is shown in the red at the top, compared to individuals without STIs who are shown in the blue. And what we also saw was that even when we used dimensionality reduction methods, MDS plots and those kinds of things, we saw that we had very distinct proteomes between from individuals who had asymptomatic STIs and individuals who did not. And we found over 400 host proteins that were significantly differentially expressed by these asymptomatic STIs. And so the gene ontology enrichment analysis that we did showed a couple of themes that came up that were enriched in the proteomes of individuals with asymptomatic STIs. And in particular, the ones that we found quite interesting were these functional enrichment of the gene ontology terms involving IL-12 in asymptomatic STI proteomes. And so what I'm showing here is these web plots, as we sometimes call them, but which really show the protein expression or the protein intensity of various proteins. These numbers here denote different proteins, and so what we see is that proteins that were which had a role, which have a phenotype, which have a function in the cellular response, interleukin-12, were significantly increased in the proteomes of individuals who had asymptomatic STIs, shown in the red here, and the individuals without are shown in the blue. And these gene ontology enrichments, the p-values were quite significant in the range between 1.4 to 1.8 times 10 to the minus 4. But we also found these neutrophil degranulation and activation gene ontologies, and following with that, interleukin-12 mediated signaling pathway proteins upregulated in the foreskins of men with asymptomatic STI. And some of these proteins which we found were PICARD, which is the apoptosis-associated speck-like protein containing a card, which is quite interesting, involved in inflammation, and so several different proteins, but really the gene ontologies, what the holistically came and showed was that these myeloid cell activation involved in the immune response was the main phenotype enriched in these asymptomatic STIs. And so what we're thinking is that so these asymptomatic STIs drive this pro-inflammatory signature, which is mediated by IL-12, possibly in the male genital tract. And we do understand that IL-12 is, from your immunology textbook days, you would remember that this is involved in the priming of naive CD4 T cells to the polarization of Th1 cells. And indeed, these Th1 cells are shown, they express CD4 and CFAR, which are HIV factors that are required for infection. And when we looked in these foreskin proteomes using flow cytometry, indeed, we do see that there are higher concentrations of cells that have this Th1 polarization. And so what is quite interesting here is, and this is work actually that also using this really proteomic approach also agrees with data by other researchers, which has shown that CD4 and CCR5 positive C cells are enriched in the foreskin itself compared to in the blood. But what we're seeing here is that asymptomatic STIs increase this even further, and we're seeing this potential IL-12 pro-inflammatory signature really. And what is interesting is that this chlamydia, which is an intracellular bacterium, right, which infects epithelial cells in the urethra, actually has this huge impact in the foreskin proteome, as most of the individuals who we tested had chlamydia infections. And so what we saw conversely in these individuals, the gene ontology terms enriched in the proteomes of individuals without asymptomatic STIs, were these proteins associated with barrier function. And yes, and so barrier function really, in our case, what we saw proteins that are involved in homotypic cell-to-cell adhesion. So proteins that are involved in the adhesion of keratinocytes to another keratinocyte were largely increased in the foreskin proteomes of men without asymptomatic STIs, and which also mirrors quite nicely work that has been seen in a Peruvian cohort, which Maria worked on, which showed the presence of tight junctions being depleted in individuals with all different types of STIs. And so really what we're seeing is that this enrichment of gene ontology terms that are associated with increased barrier function in men without STIs. And so the other really interesting part of the data, which I don't have time to show now, is also we saw several proteins that are sequestered by the virus that were also upregulated in the proteomes of men with asymptomatic STIs. And so we think that it's a combination of all these factors, these multimodal effects that asymptomatics have on susceptibility in the male genital tract, where the host proteome is there's a dysregulation there, and there's also in the microbial proteome, which unfortunately I don't have time to show the data that we see there, but we also see significant differences between the proteomes of men with asymptomatic STIs and without. And then we also see these cellular factors coming into play here with these myeloid cell activation and recruitment of neutrophils into those individuals. And so what we then went on to do was so we're seeing this role of myeloid cells that were coming up. And so with that, we then decided to really try and investigate what these myeloid cellular factors that are associated with HIV susceptibility might be. And so the question was really, what role do myeloid cell subsets play in HIV acquisition? And myeloid cells have recently, been taking center stage in the HIV field because they've been largely ignored as they were thought to be not HIV infected, but we have seen convincingly with data from various labs, including the Geno, including from the laboratory in France, showing that that have actually shown us that macrophages within the tissue itself, these were shown in the urethral macrophages are infected by HIV and form the reservoir. And so, macrophages, which are myeloid cells, have really been shown to be playing quite a prominent role in these and other studies. And so we wanted to look at what role myeloid cells particularly play within the foreskin tissue. And so to do this, what we did was we developed an ex vivo HIV infection model, and this was work done by a talented PhD student who's now a postdoc in the USA, Bokani Lea, who worked on developing this ex vivo HIV infection model, in which what we did there, we've isolated foreskin cells that were obtained from foreskin from men undergoing voluntary medical male circumcision. And these foreskin cells were isolated firstly from the epidermal sheets that are part of the foreskin. And the reason why we chose epidermal sheets is because this is where the majority of the immune subsets are, immune cells are. And what we used was this really fortuitous phenomenon in which we find that when you put epidermal sheets in culture media, the immune cells spontaneously migrate out. The reason why they spontaneously migrate out, were they primed already for movement when they were still in the body or not? But what we find is that these cells spontaneously migrate out of the epithelia. But what is really cool about it is when you digest the foreskin tissue or these epithelial sheets, the viability and the state of the cells that you get is quite depleted. It's particularly myeloid cells. They're very fragile cells, if you would say, and most cell isolation methods are not favorable for them. And but we found that this method in which we really just leave the foreskin to spontaneously migrate out allows us to isolate cells that are viable. And interestingly enough, it's predominantly immune cells that migrate out and not the keratinocytes. And what Bokani worked on was she optimized the MOI that could confer the best detection of viral infection, but without using copious amounts that are not physiologically relevant. And so we used an MOI of 0.01, which really is quite physiologically relevant to infect the cells isolated from the foreskins. And we looked at this infection over time, and we detected the presence of infection using flow cytometry, in which we looked at the presence of P24 or the expression of reporter marker genes. Okay, and so this is what Bokani's gating strategy for her flow cytometry panel was like. And so what we find is that from the foreskin that spontaneously migrate out, you are able to isolate, if you do your flow plotting well, you can isolate both the myeloid cell fraction and the lymphoid cell fraction, and actually immunophenotype them simultaneously. And so we were interested in looking at Langerhans cells And we also were interested in looking at macrophage-like cells. We will not speak about the T cell data, but the T cells are also present in these cell fractions that we immunophenotype and subsequently infect. And using this gating strategy, we were able to isolate Langerhans cells, which are defined as CD1A and CD207 positive cells. And then from there on, we then, from the negative Langerhans cell fraction, then looked at the expression of CD11C and CD14 and looked at those markers on Langerhans cells, and then used those markers to really delve into the macrophage populations. And we were interested in looking at this concept of macrophage polarization, although we understand that macrophage polarization really is not a, it's more like a continuum rather than a, extreme. But we wanted to understand what the role of the different myeloid, macrophage states would be with regards to HIV susceptibility in an ex vivo infection model. And we also looked at CD169 macrophages and looked at the M1 and M2 intermediates, which we'll show here. And so what we used, the viruses that we used for ex vivo infections were from collaborators that we work with, Frank Kirchhoff at the University of Ulm, and Professor David Russell at Cornell University in Ithaca. And what we obtained from them were these reported strains of HIV that are expressing either M-cherry or GFP. And what we then were interested in, as I mentioned, was to look at the impact of viral tropism in ex vivo infection as well. And so we had laboratory engineered strains of HIV, which are R5 tropic, as well as the X4 normal trope that we use, and subtype B, transmitted founder strains that we obtained from Frank Kirchhoff, that originally came from Beatrice Hahn's lab, but which were engineered in his lab to express GFP. And our aim really was to look at GFP and P24 expression to really identify infected cells, and also looked at subtype C, transmitted founder strains, as well as chronic IMCs isolated from individuals with chronic infection. And so we infected our foreskin cells with the different viral constructs and looked at the levels of infection in the various immune subsets. And so what we found is we saw that in the majority of foreskin cells that were present in these foreskins, really in individuals which are HIV negative, and in these individuals without asymptomatic STIs, the majority of these cells are have this M2 phenotype. And we found this consistently, whether you look at foreskins that are whether you look at the inner part of the foreskin or whether you look at the outer part of the foreskin, we really saw that these M2 macrophages are the majority of the cells. And we saw, because we were able to quantify within the same cell fraction, the Langerhans cells, we were able to compare the numbers of Langerhans cells and macrophages. And as expected, Langerhans cells are a rare cell population. They are much fewer than macrophage cell subsets. And as shown here, but what we found quite interesting was that there were all these different types of Langerhans cells. I mean, Langerhans cells have been previously regarded as a very homogeneous cell type, but we see in our data here that actually they're very heterogeneous cell type with the traditional Langerhans cells that have been described. But we've been seeing some very interesting markers being expressed on Langerhans cells, such as CD14, also we identified these unconventional cells, and we call these unconventional macrophage-like cells, which are expressing combinations of these markers, which using normal immune paradigms, we really couldn't classify into what subset. But in our case, we weren't too bothered about it unless they were infected by HIV. So we then investigated the impact of ex vivo infection on these various immune subsets. And before we did that, what we wanted to do was to really establish whether the infection we were seeing ex vivo was real or not. And in order to do so, we looked at the inhibition of infection using Maraviroc, and indeed we saw that the P24 expression that we're seeing in our HIV-infected cells was significantly reduced in the presence of Maraviroc. And so Maraviroc, which is the CFAR5 blocker, and ARV, which is used, significantly decreased this infection, which led us to have to really understand that this is biologically relevant infection. And we also, when we looked at the impact of infection over time, and this was done over 5 days, what we saw in the lymphoid foreskin cells, we saw this increase in the P24 signatures, and in myeloid cells, a steady increase, but much less as expected in the myeloid cell fraction, but significantly present. And so we also then saw the impact of different viral subsets, which I spoke about earlier, and I'll speak more about these at the end of the talk. But what we found was that, and as expected, well, in vivo, we do understand that HIV infection, as I mentioned, there are individuals who do not get infected, even though they're highly exposed. And even in our hands, in our ex vivo model, we find that there are some individuals who, no matter what we do in the same experiments that we do with other individuals, who will just not be infected. Their cells do not get infected. And even when we go at higher MOIs, they just do not. And we think this is very interesting, and we're quite interested in following this up. And this is work that we're quite keen to try and understand what the genetic factors that might be present that could confer this, that are other than the known ones that are there, but on a cellular level. And so what we definitely see is that we see a variable infection in different individuals, which are shown here, different participants, like this participant here, number 34, just very low infection, and yet the others, you find quite decent amounts of infection. And so what we then wanted to do was to just really to get into the depths of these immune subsets. As I mentioned, we really found that our manual gating strategies resulted in more questions than answers. And so we decided to take a computational approach, which is an unbiased approach, to try and understand this multiparametric flow cytometry data that we generated. And so in order to do this, this is work that we're doing in collaboration with Dr. Sonwabile Zanibe, who's also based at the University of Cape Town. And this is analysis that we conduct on our flow cytometry files, the FCS files that we get from our normal flow data. So rather than using pseudo color plots, try and understand what's going on, where you have to look at one marker at a time, one antigen at a time, where you can actually look at multiple markers all at the same time. And of course, no human can do that, and hence the use of the computational methods. And we use this self-organizing map to allow the data itself to tell us what the clustering, what the different sets of different subsets and cell states that are present in these foreskin cells. And so this flowsum is a visualization aid that allows you to simultaneously look at multiple markers all at the same time, and really in an unbiased way find different subsets that are present in your fractions. And we use this analysis to try and understand the differential infection of ex vivo infection in our hands. And this is what we saw. I know the slide takes a bit to load because it's quite a high. It has lots of pictures on it. And so this is how we conducted this ex vivo HIV infected macrophage-like computational analysis. We used our manual gating to isolate our CD11C positive or CD14 positive cells. And then from that fraction, we found the cells that are expressing P24 in our infected ex vivo infected experiments, and compared those to our HIV negative fractions. And then we combined the data, and using various data pre-processing pipelines in R, we then look at the self-organizing maps, and the heat maps are then tell you what the expression of each marker is and its extent, and then you end up with these clusters of cells that you can then identify that are there. And so what we found with our macrophage subsets, which is what I'm showing you today, we found we've conducted this analysis on Langerhans cells and lymphoid cells as well. So what you do is you use these expression data that you get from your flow plot and your MFI data to really see the expression of your different markers, which we think is really, really, really cool. And then you combine it together in these multidimensional reducing plots and these t-SNE plots, which then using FlowSOM tells you the best number of clusters that best differentiates your data. And to use this, we use various tools that are very similar in single-cell RNA analysis, which, the Oval criteria to determine the optimum number of clusters. And what we found in our hands was that our macrophage subsets were best clustered into 12 clusters, and these are the 12 clusters that we're able to identify from our data that came from our macrophage subsets. And so from the data that you get from the MFI data as well as expression analysis, you can then determine what antigens are being expressed on those particular cells. There we go. I'm just waiting for the slide to load. And so what we saw is we were then able to identify differentially infected macrophage cell subset. And what I'm showing here, we found one cluster of macrophage subsets which were more frequent in the foreskins that we infected with HIV ex vivo. And we saw in those same foreskins, in this case, I'm showing the 3 that were significantly differentially infected in uninfected foreskins. And so when you compare this uninfected foreskin plots as compared to the infected ones, what we saw was that the frequencies of certain clusters, in this case, this blue cluster here, was significantly increased in the uninfected foreskin cells. And this cluster here, B, was significantly increased in the in ex vivo infected foreskins. And so the analysis that we then do is when you compare the uninfected cells, compare the frequencies of those clusters within the cells that in your ex vivo infected shown here in this brown compared to the uninfected, you're able to see these differences. And I'm only showing the ones that are significantly differentially expressed. In this case, the subsets that are differential between uninfected and infected. And these are the clusters that we see there. And then you're then able to try and really find out what antigens that these cells are expressing. And that will load up just now, I hope. Yes. And what we saw was that we found that in our uninfected macrophages, which we were quite surprised by, we found these CD206 positive, the mannose receptor, CD11C positive. There was CD40 negative, but also CD45 negative, which we were quite surprised by. We found that these subsets were more frequent in HIV uninfected macrophages. And we found that the differences between these different clusters, F, B, and C, were to do with HLA-DR expression. And this small cluster here, D, was the only cluster that we found in the cells that were more in the uninfected foreskins that was CD45 positive. The others were actually CD45 negative. And this is where we see that this cluster here, when you look at the CD45 expression, it's really quite bright here, whereas the other clusters were CD45 low to negative expressing cells. And so we would like to interrogate this a bit further. And what we have seen when we're using manual gating, this data is also corroborated. And as I mentioned, this analysis also can be done using the various subsets that are present and in the Langerhans cells as well as the lymphoid cells. And so in summary, what we saw this differential ex vivo HIV infection of various subsets and cell states. And we saw that these cells that were not infected with these CD206 positive, CD11C positive, CD40 negative, CD45 negative, they were expressing CD4 and CCR5 as well. So the other markers, they're also expressing them, except for HLA-DR from the other one, which we found quite, which I didn't show there. And then I'm just going to speed along and show you the last part of our data, which is the impact of viral tropism. And this is really looking at a cellular, a viral factor, which does have a role with the host factor. As we understand, receptor usage is really the reason for this viral tropism. And so trying to understand what viral tropism, what its impact is in HIV susceptibility in these ex vivo infected cells. And so to do that, we have used these foreskin biopsies, and we have infected them with these various virions that are subtype C, D, and B that are expressing the nanoluciferase reporter gene, which we obtained from Christina Ochsenbauer at the University of Alabama. And we infect these in our foreskin cells, and using our usual flow cytometry-based methods, we identify the cells, and also we using, which is quite nice with this nanoluciferase releasing cells, reporters viruses, we can look at the supernatant and see the extent of virus production and virus release. And this is work that we're currently doing, and in collaboration with the viral IMCs, as I mentioned, were obtained from Christina Ochsenbauer, and Gabrielle, who is from the University of Cornell, has been conducting this work together with Shival Sukram, who is a PhD student in my lab currently. And I will not go into the details of the construction of these IMCs, but the important thing to note is that we use these viral IMCs to infect human monocyte-derived macrophages, and as well as foreskin cells, which we use the subtype D viruses, and looked at the release of nanoluciferase, and as well as looked at alamo-blue to see the impact of cell viability. And so what we saw really was that in the human monocyte-derived macrophages, and this is really data that is quite fresh, and so we're still increasing, our power. We've seen definitely the impact of drugs on viral tropism. So not all viruses, the amount of nanoluciferase that is released from these different IMCs is very dependent on the viral trope that is used. And this could be due to various reasons. Is it biological? Is it not? And the kind of questions that we're really interrogating. And what is the impact of drug on this? And we do start to see that actually some drugs, in this case we use efavirenz, the drug Tagrivier, have a differential inhibition of infection in human monocyte-derived macrophages. And in our foreskin cells, we're still really working on this, but we're definitely seeing an impact of viral tropism and an impact of drug inhibition by viral tropism. And so in our data, which was also generated by Bukani, we also have seen that viral tropism, as would be expected, it has an impact. And when you're looking at your lymphocytes, your CD3, CD45 positive CD4 cells, as well as your myeloid cells, certain viruses infect certain cells better. And so we're really interested in interrogating this. And so in order to summarize this, this is a slide that I got from one of my students suggested. He said to me, I said to him, how do I wrap all of this together? And he said, you need the Spider-Man meme. And here it is, indeed. What is it? Which factor is it that really we could use to pinpoint what it is that really has an impact as a host factor to an impact on HIV susceptibility and a correlate of infection? And really it's the combination of all of them. Viral factors play an important role. cellular factors, as we've seen, definitely play a role, different subsets, the ones that are present. And we've also seen the molecular factors from our proteomics data. And so with that, really, we've seen a combination of these different things that provide either this HIV susceptible phenotype or this antiviral phenotype and involve these multiple things. And so really there's a need for this multi-omic kind of analysis to really look at these. And future studies that we're interested in looking at would be looking at the impact of myeloid ontogeny, or even looking at the impact of what do we see in the foreskins of men who are on ARVs? Do we find a dysbiosis in their cell subsets and those kinds of things? What about immune factors? What about the immunogenomic factors? And so with that, so that I can have some time for questions, which I would love to hear from you, I would like to thank the various people that have been in our lab, my collaborators, particularly my K43 collaborators, Professor Clive Gray, who's at the University of Stellenbosch currently, and David Russell in Ithaca, and Frank Kirchhoff in Germany, and colleagues at the University of Cape Town. I would like to say thank you, and of course, my funders, and take any questions.